let's talk about where the data comes from. Because there's no bioinformatics without some bioinformation, right? So we'll start with all that DNA information and RNA information. Now, sometimes I'm just going to say DNA when I mean nucleic acid, because there are things like coronaviruses that only have RNA. They never have DNA, but they still have genes and all the rest of that. So put that aside, and yeah, let's talk turkey. So, to really get this, you've got to go into a little bit of the molecular biology, but I think it's neat because it's going to answer some questions that you may have. You've probably seen a sequence, but I doubt you've ever actually generated a sequence. Yeah, a lot of people haven't, even in the business. So, this is how it works. Yeah. And at the very least, it's a very long, interesting story that you can tell around next year's Thanksgiving table. Okay, so sequencing, as it is done in the modern way, usually revolves around this guy, which is the enzyme Taq polymerase, spelled TAQ. It's an abbreviation for, I think, Thermus aquaticus or something very close to that. It's the name of a particular bacterium that it comes from. What it does is it is going to take a piece of DNA, and if you just give it one piece of DNA, it can't do anything. But if you give it that piece of DNA and another smaller piece of DNA that's bound to the end of it, it will extend that DNA until it runs out of things to copy. And so in this way, you can make a copy of whatever it is. And basically, most of our sequencing techniques revolve around the act of copying. Yeah. We make a bunch of copies of it, and then we can get the sequence from it. Mostly, this is because a single piece of DNA is incredibly tiny. You can't find it if you were to go and look for it. Like put it in solution and then keep moving around your electron microscope. You will never find it, or it'll take a while anyway. Um, but if we make millions, billions, trillions, actually numbers much larger than a trillion copies of it, then all of a sudden it's like a substance. It's a thing you can see. You can hold it. It has enough mass that we could detect it with sensitive instruments. So this is why we use polymerases, because they turn a few things into a lot of things. And then once we have a lot of things, we have a lot more options. Yeah. All right. So, yeah, Taq polymerase. It'll copy DNA if you give it DNA to copy. And the nice thing about Taq is it doesn't care which DNA you give it. You can give it any old gene from any organism, people, jellyfish, whatever. A gene you've put together yourself from oligonucleotides, and it will copy it as long as you also give it a little primer that can bind to the end. And so primer is just a small piece of DNA that is going to be reverse complementary to the template. So that means if the template has a C nucleotide, then the primer's got to have a G nucleotide so it can bind to it. Yeah, but that's all like Bio 1 from years ago, right? Yeah, so we don't need to cover this. If you do need a refresher on it, there's a lot of good videos on YouTube. Search PCR tutorial and, go wild. There's plenty of them there. And you'll be fully refreshed. Yeah. All right. Okay. So our super quick PCR refresher. There we go. To make PCR work, you need a bunch of things. Now, I'm only going through this because it's going to be important later. This is like one of those mystery books where, these are the clues that you're going to put together, except I'm telling you right now, these are clues. Look at them. Yeah. Look at the clues. Okay, so you're going to need a polymerase, which is an enzyme that can join single nucleotides into long strings of nucleotides, given the right conditions. What are the right conditions? Oh my gosh. Yeah, it has a very long list of demands. Yeah, longer than, Taylor Swift at her average concert. All right, so here we go. So you've got to have nucleotides. Otherwise, it can't just generate DNA out of thin air. It's got to actually attach nucleotides together. So you give it what we call deoxynucleotides. Those are DNA nucleotides. If we just see NTP, like nucleotide triphosphate, that means it's probably going to be the RNA version, or that means somebody was sloppy and didn't put the D there. But the D means that it's definitely DNA. And so like the ATP that you would use as energy is the ribose version. So that would just be ATP. And deoxy ATP is the one that you use when there's a deoxy T in the nucleotide sequence you're trying to copy. Then you put in an A. And so there you go. That's how you make DNA. Oligonucleotides, we also call these primers, and they are complementary to the sequence that you want to bind. And so to do PCR properly, and yeah, you should do it properly, you want two of these. One's going to be facing this way, and it's going to be at the start of the gene you want to amplify. The other is going to be facing this way, by which I mean it's the reverse complement of the sequence you want to amplify, and it's going to be down at the other end. And so if you were to extend these, they would cover from the start to the end of the thing you want to amplify. And if you put those in there, PCR will generally work. The other thing that you need for PCR is a little bit of magnesium. And so expert PCR jockeys will say, well, if you adjusted the magnesium concentration, if your PCR doesn't work, and to tell you the truth, yeah, it helps. The enzyme, the polymerase enzyme, holds on to magnesium atoms, and they are kind of loosely held. It'll grab one, then it'll fall out, and it'll have to be replaced by another. So if you put in lots and lots of magnesium ions, then basically the polymerase kind of never stops, and it'll make mistakes and just keep going, really good in some walks of life. I'm told football cornerbacks need to be like that. But for a polymerase, if you needed to actually make an accurate copy, you want it to go a little slower, and you want there to be time for the error correcting machinery to recognize a problem has happened before it's already, 10 nucleotides downstream, and there's nothing you can do about it. All right, so adding a ton of magnesium will do that. Adding a tiny bit of magnesium will really slow things down. And sometimes you can get a PCR product if you add just a little more, or sometimes you can make some of the extra PCR products go away if you add a little bit less. That's the trick. And then you need a template, the DNA to be copied. Yeah, that's as basic as it gets. So it's got 3 steps. These are the same 3 that you've always learned. First step, we get it really hot, about 95, a little less than boiling. It's going to split our double-stranded DNA into 2 beautiful single strands. Second, we're going to cool it right down to 50 degrees Celsius, which is still really hot for humans, but yeah, it's just fine for DNA. We cool it down to somewhere between 50 and 65 by calculating what's the temperature where our oligo, where our primer should actually bind to its complementary sequence on the template. And you can calculate this online. There's little melting temperature calculators, and they'll tell you exactly what temperature you should use. And then you try a range, and it usually works. And then extension. The PCR enzyme, the Taq polymerase, or whichever one you're using, is usually at its best between around 68 and 72 degrees. Different manufacturers will tell you different things for exactly the same enzyme. They're just trying to optimize for either fewer mistakes or faster speed or something like that, basically. So yeah, there you go. That's PCR in a little nutshell. And let's show you the second slide, which is just summing it all up. So PCR is polymerase chain reaction. So the chain reaction is that you do those 3 steps, denature, anneal, extend, over and over and over again. So the first round, you'll take 1 strand and you turn it into 2. Second round, you take 2 strands, you turn it into 4. Third round, you're going to take 4 strands, you're going to turn them into 8, etc. I'm out of fingers, but you get the idea. And so you can go from very few strands to a actually ridiculous number if you run it through all 40 cycles. Yeah. And you can do that math yourself. What's 2 to the power of 40? Yeah. And you'll be pleasantly surprised how large the number is. Yeah, so that's the basic idea. That's PCR, and that's the first step. So I'm going to break this into steps. This is step 1. Get ready for step 2. It's just around the corner.